Thank you. My name is Katherine Lake, he mentioned, and this summer I had the wonderful opportunity to study the regulation of apple acidity in the Paneros lab. So my project looked at the ALMT transporter family that moves anions across the membrane. ALMT stands for aluminum activated malate transport. And this family plays important roles in plant mineral nutrition processes, responses to abiotic stress, and my focus, fruit quality. In the case of apple, this plays out in fruit acidity and taste. The Chang lab here at Cornell has identified two ALMT alleles that encode transporters that determine fruit acidity. Collaborating with this lab, we have determined that the alleles differ in the fact that one encodes a protein that is lacking part of the C terminal, which we can see in this image above. Genetic studies have found that this shortened ALMT transporter causes apples to taste sweeter than the more acidic counterpart. Additionally, previous work has shown differences in transport capacity, which we can see here in this middle figure. And additionally, we can see a decrease in transient expression for the truncated transporter compared to the full-length transporter. I confirmed that this is also true for wheat ALMT, but today I will be focusing on the apple. So this leads me to my question. Does the truncation of the ALMT transporter affect protein turnover as a method of regulation? So to answer this question, I use tandem protein fluorescent timers, which I attach to my transporters. The timers consist of two fluorophores, a GFP and an M-cherry. The M-cherry matures at a slower rate than the GFP. And so, as a result, we can find the ratio of the intensity of M-cherry to GFP to find the relative lifetime of the transporters. A greater M-cherry to GFP intensity means that the protein population is relatively older. And if the ratio is smaller, then it means that the protein population is relatively younger. And so, in order to add this fluorescent timer, I use Greengate cloning, a two-step process that combines the protein and the timer in order to express the construct in tobacco. This figure here summarizes a lot of my work this summer. So we have multiple panels. Here in the rows, we have the full-length ALMT, and then here on the bottom, we have the truncated. In the columns, we have the two fluorophores that I mentioned earlier, the GFP and the M-cherry. So right off the bat, we can see several observations. We see the decrease in transient expression that we saw earlier. And we also see more punctuated expression in the truncated compared to the GFP. And so earlier I talked about the ratio. So to find that ratio, I plotted the M-cherry as a function of GFP intensity for every single pixel in these images on the left. And I found the slope, which is equal to the ratio. And so we can see a greater slope for the truncated transporter compared to the full length. And so to quantify this for many cells, I made a bar graph. And so we can see here a larger ratio for the truncated. So this means that the truncated transporter has greater population lifetime compared to the full length. Consequently, we can conclude that the truncation does in fact result in differences in protein turnover. Moving forward, research will begin to hone in on what aspect of the C terminus is playing a role in regulation. Furthermore, using this system, we will be able to build off of additional work that I did this summer which resulted in preliminary results that show that phosphorylation also plays a role in determining protein lifetime. So in conclusion, our fluorescent timer system allows us to study both the role of the C terminus and the effects of post-translational modifications on apple acidity. I would like to thank Miguel, Nathan, and the rest of the Pinneros lab for mentoring me this summer. Dr. Marcus Wirtz at the University of Heidelberg for sharing the GreenGate cloning system with us, and Olivia, Delaney, and George for organizing this internship program. Thank you.
Hello, everyone. Thank you, George, for introducing me. I am an REU student from Minnesota working in the stress architecture and RNA biology lab. So this summer, I have essentially been working on 2 projects with long non-coding RNAs. To give you a little bit of background, long non-coding RNAs were once considered insignificant portions of the eukaryotic genome, but recently the identification and functional annotation of lincRNAs has been a budding area of research. This is due to their implications for significant roles in stress responses in plants, as well as other functions. So they're known for their short open reading frames, lack of coding potential, their length greater than 200 nucleotides, and they are also poorly conserved. So as I said, I've sort of been working on 2 different projects. One of my projects has been with sorghum, also known as broomcorn, and then the other one has been with Arabidopsis, which is a model organism for plant research and like plant genetic research. So there are kind of 3 steps to the process I've been working on this summer. So the first step is taking mass amounts of public available RNA sequencing data and then running it through a program called RMTA, which does read mapping and transcriptome analysis. So it essentially takes this RNA sequencing data and maps it back to a reference genome. And then from there, we can run the output from RMTA into a pipeline called EvoLink, which will then identify lincRNAs, where we can use those to make some functional predictions, look at their expression, their conservation, and then perform more analyses on them. So then, as I said, step 3 is essentially screening through these different plants that are possibly mutated or something like that to look at their lincRNAs. So this summer in sorghum, we've started the beginning of this process. So essentially step 1, there has been approximately 2,500 sets of RNA sequencing data that's been sorted through, and approximately 85,935 lincRNAs were identified. And so here you can see that this is a expression map that, anyway, you can see it. Basically, these are the 10 different chromosomes that are on sorghum. And then with our experiment for Arabidopsis, we have screened through approximately 155 plants. And then, so these plants are mutated with tDNA insertions that are targeting lincRNAs. And we grew these in control conditions to look at any sort of growth effects or delays. We essentially expected there to not be any significant difference in growth rate because lincRNAs are so prevalent in stress responses. And we were correct in that they had no significant difference in growth rate for 11 of the 17 mutated plants that we've grown. However, as you can see here, there were slight significant differences in 5 out of the 17 plants that we've screened through. And so we're going to use those as potential candidates for further study and grow them using more replicates. So, sorry, it's hard with the Zoom. I'd like to talk about what's next for this experiment as well. So in sorghum, they're going to be ran through the rest of the sequencing data is going to be continued to run through RMTA and EvoLink. And then the lab will continue to run a series of experiments to hopefully identify the functions of its lincRNAs, and then also run them through stress experiments. And then for Arabidopsis, there are going to be about 42 more genotypes grown under controlled conditions. And then, as I said, we will repeat this with the interesting candidates and examine them for any disruptions in development. And essentially, the goal of this project is to fill in this gap of knowledge about lincRNAs in these agriculturally significant plants. And so basically, this will help develop plants with better responses to a variable climate and hopefully improve the selection for more resilient plants in the future. I would like to thank the Nelson Lab, everybody else in the stress architecture and RNA biology lab, as well as Cornell and everyone at BTI. Thank you very much.
All right. Hello, everyone. My name is Holly Carter, and I've been working in Dr. Andrew Nelson's lab this summer. And my project is on high throughput phenotyping of LINC-COBRA1 and DXO1 mutants in Arabidopsis plants. So as you know, phenotyping is a very important tool that all scientists use to study different genetic factors that might contribute to different biological processes. So improvements on these phenotypic tools can be very helpful for scientists to gather larger data collections, gather better data analysis, things like that. So my project will be using PlantCV software and further data analysis pipelines we've developed in order to more accurately quantify our plant phenotypes. So in my experiment, we used Arabidopsis plants and 3 different types of mutants, which includes the first one, which is a reduction of LINC-COBRA1 mutant, LINC-COBRA1 expression. The second one being a complete deletion of the LINC-COBRA1 related gene, so there'll be no expression in it. And then the third mutant we'll be looking at is a DXO1 mutant, this one here, which is, so past research has found that the DXO1 mutant has very visually obvious stunted plant growth between it and the wild type, when the LINC-COBRA1 mutants that we're looking at have a very subtle difference that we can kind of see, but can't really visualize it as much. So that's why we took the fresh weight of them in past studies. Thank you, Kramer. So for my experiment, we're more interested in the growth rate throughout the entire experiment, not just looking at certain instances like these down here. So in order to do that, we developed, I say we, I mean the Jalauskas and Nelson Lab developed phenotypic rigs that have 2 Raspberry Pi computer camera setups in each rig, which will be used to take images every 30 minutes for when the plants are growing in the rigs. And then we'll collect those images over about a 2-week period, and then use PlantCV to analyze these, as seen here. The PlantCV will be able to give us the area of each of these plants for each of the images in pixels, which we then used to create some graphs. So here we see the plot that has a leaf area in pixels for the y-axis, and then total minutes of the experiment for the x-axis. And here we have the plants divided by the rig and camera it was in. This was because we realized that there was a significant difference in the growth rate between our rigs, caused by things like positional effects, so where they were in relation to each other. And so we normalized the leaf area and growth rate in order to get the graphs that we really are interested in, which is here. So here we have the normalized growth rates between all the different genotypes, so like the mutants versus its wild types. Here we can see that shown with these little stars here, I hope you can see this, that there's a very subtle difference for the first link cobra 1 mutant at one point in time, like certain point in the experiment. So whenever we see the second link cobra mutant, we can see that there's a little bit more of a significant difference throughout the experiment. And this is likely due to genetic redundancy, because the link cobra 1s is just a class of multiple cobra link RNAs. So we are able to visualize the subtle differences. And of course, here we can see in this last graph, the DXO1 mutant has a very obvious difference, and that was found to be due to up and down regulation of specific genes that influence the plant growth. So further improvements on this, on our project in the future will have to do with being able to look at different types of plants and phenotypes using our tools, along with a more user-friendly interface, so multiple plant scientists can use our analytical pipeline. Thank you. And I want to thank everyone who helped make this program possible this summer.
Thank you. Yeah, my project is titled The Effects of Withanolides on Insect Herbivory. I have chopped up my poster to make it easier to see. So plants and herbivorous insects co-evolved in what is known as an evolutionary arms race, where they develop adaptations and counter-adaptations to each other over time. Often plant defenses against insect herbivory are mediated through secondary metabolites, which are phytochemicals not involved in basic life functions. Secondary metabolites are highly diverse and many have not yet been fully studied, including a group of steroids called withanolides. Withanolides are primarily produced by members of the Physalis genus, which is in the Solanaceae family. It includes the crops of tomatillo, ground cherry, and goldenberry. If you haven't really heard about these, I wouldn't be surprised because they're not really grown in New York, and this research is part of an effort to make the Physalis crop plants more popular among local farmers to increase agricultural and economic diversity. Existing literature suggests withanolides reduce insect herbivory and performance, and by better understanding their function, we may be able to harness their anti-herbivory effects by breeding for withanolide production within Physalis species, or applying them as pesticides. In my study, we tested the hypothesis that the presence of withanolides on plant tissue reduce insect herbivory and performance through reduced leaf area consumption and reduced insect weight gain. To test the effects of withanolides on herbivore feeding, we fed herbivores leaves with and without withanolides. We did this by running a 24-hour no-choice bioassay consisting of a leaf disc and an insect in a sealed petri dish. For insect, we used the cabbage looper larva. The scientific name is teeny. because they are generalist agricultural pests that have previously been shown to be susceptible to withanolides. For the leaf discs, we collected leaves from potato plants, as they are a common crop plant that teenie readily consumes. Each leaf disc had one of 3 treatments applied, either a withanolide treatment, which is extracted in ethanol as a solvent, or an ethanol treatment to control for the solvent, or a no treatment to control for any ethanol effects. Some leaf discs were left without larvae to control for leaf necrosis, as previous experiments have shown discs to rot over the 24 hours. So for you to get a sense of what we saw, the untreated disc after 24 hours looked quite nice. The withanolides have a dark pigmentation, which you can see there. And then you can see some necrosis on the bottom image, and you can see a rather hungry and messy larva on the leaf disc. So we found that our results support our hypothesis. Plant tissue treated with withanolides had reduced insect herbivory and performance. For leaf consumption, which is the graph shown here, larvae on withanolide treated leaf discs consumed less tissue compared to the ethanol control. On average, larvae on the ethanol control ate 4 times as much leaf compared to the treatment, which suggests withanolides reduce leaf palatability. There was also a significant ethanol effect, since larvae on the untreated discs ate 60% more than the ethanol treatment. For larval weight, larvae on the withanolide treated leaf discs lost weight. So that faint gray line is the average starting weight, around 15 milligrams. On the treatment, they lost weight, around 3 milligrams, while those on the ethanol control gained a small amount of weight, around 0.1 milligrams. The ethanol control larvae then weighed 23% more, which suggests withanolides reduce herbivore performance. We also measured some other effects shown here. Larvae on withanolide treated leaves were more likely to be in poor health, which is defined as being dead, dying, or lethargic, and there was no ethanol effect found. They were also far more likely to be found off the leaf, as you can see on figure 4. No caterpillars on withanolide treatment were found on the leaf. There was no ethanol effect once again. And finally, we found that the presence of ethanol had an effect on the leaf tissue, increasing chances of necrosis compared to the untreated tissue. We don't know if the effects of the ethanol we observed on herbivory are then indirectly or directly mediated through the necrotic tissue. In terms of future research, there is a lot to be done. Unpublished work from our lab suggests the effects of withanolides are not consistent across species, indicating our hypothesis may require modification. We hope to look into more species, as well as to try to identify the mechanism by which withanolides affect insects. Thank you. I would like to thank my mentor, Julie Davis, and our lab leader, Jennifer Thaler, as well as Savannah Deo and Christophe Duplat for helping me in my research. Thank you.
Hello. Thank you all for sticking around. Sorry, we'll get to the presentation mode here. Okay, so my research this summer was on the role of stress granules in regulating growth in Arabidopsis. And as George said, I worked in the Nelson, Jalowska, and Skier's lab. Dr. Manny Saluja was my mentor, and an amazing one at that. So a little bit of background, we were looking at stress granule formation. Sorry, I was on the wrong slide, there we go. We were looking at stress granule formation under heat and salt stress in Arabidopsis. Previously, we had discovered that the ecotype C24 had a differing growth rate under salt stress. So when we looked at these Arabidopsis, we chose 2 different ecotypes, Columbia and C24. These ecotypes were overexpressed to transformed to overexpress RBP47, which is a red fluorescent dye, and that is how we saw our stress granules. So as you can see right here, those little bright red dots are our stress granules. Before I got here, Dr. Saluja performed an experiment in liquid culture to see when stress granules formed. We found that 37 degrees is a good temperature to see stress granule formation under heat stress, and that 125 millimolar salt concentration was ideal to see salt stress in both Columbia and C24. So my first project was to look at stress granule formation on MS plates because we wanted to also monitor root growth. So I just wanted to repeat the experiment that Dr. Saluja performed in liquid culture on MS plates. So I took 6-day-old seedlings and put them in 37 degrees Celsius for either 10 or 30 minutes, and then took confocal microscopy imaging of the seedlings. And what we found was that in 30 minutes, both C24 and Columbia formed stress granules. And then for salt stress, after 6-day-old seedlings, they were transplanted onto 125 millimolar salts, MS plates with 125 millimolar salts. And then we looked at those images and we found that while Columbia formed stress granules at 4 hours, C24 did not form stress granules until 6 hours. So that we see that in salt stress, they form stress granules at different times. So then for my growth analysis, what I wanted to do was look at the growth rates on those MS plates. So every day for heat for 4 days, I scanned those plates in and used SmartRoot and ImageJ to trace the primary root. And then I did the same thing for salt stress, but those were for 7 days because Arabidopsis can withstand salt a little bit more than it can withstand heat, 37 degrees Celsius. So if you look at our graphs over here, you can see that there isn't a significant difference in C24 until the 3rd day after treatment started for heat. But for Columbia, there's a significant difference right away on the 2nd day. Since we saw stress granule formation occur at the same time for heat stress, these were not the results we were predicting. However, you can also see that our data for C24 is a little crazy. There weren't as many data points, and we think due to low germination rates for the C24 ecotype, so we think that there needs to be a repeat done on the heat experiment, and so there can't really be any conclusions drawn. There were too many errors, and we want to repeat this experiment. But for salt stress, we see a little bit more of an interesting find. In C24, there's a significant difference the 2nd day after treatment, but there's a significant difference between the control and the experimental group on the 1st day after treatment for Columbia. So based off of this, we see that when Columbia formed stress granules earlier under salt stress, we also saw a difference in main root length, in growth inhibition in Columbia before C24. So from this, we can suggest that there is a possible relationship between stress granule formation and growth inhibition under salt stress. Like I said, due to low replication and just higher variability in growth, we want to repeat this under heat stress and take a closer look at our data before we come to any conclusions on that. But yeah, that's the general gist of what I did this summer. I would like to thank Dr. Nelson, Dr. Jalowska, and Dr. Skiers for everything they did this summer. I had an amazing opportunity. And also Dr. Murphy, who is an affiliate at Centenary College of Louisiana, which is where I'm performing my undergraduate. And yeah, I had a great time. Thank you.